Good morning. Thanks for the opportunity to talk today. I just want to say again that honor that was very inspiring, I think, for all of us, and I think is really it was a great snapshot of where we are in the field of genomic medicine, which is that it's clear there's an enormous amount of work that needs to be done to make this concept a reality, and yet at the same time, there's enormous reason for optimism, and you exuded that optimism, and I have to say, I think that was inspiring because, we all worry about the concepts of genomic medicine being subject to overhype, and sometimes that results in the rest of the world giving up and saying, oh, well, you haven't worked out everything that needs to be worked out for this field, and therefore maybe that was just a fad, the concept of genomic medicine. And I feel like we're at a moment now where it's clear this is not a fad. There's a lot that needs to be worked out. There has been too much hype in certain areas, and yet we're now starting to see these ideas really pay off for patients. And so this feels like a really exciting moment. So I'm going to talk about some thoughts on where we are in cancer precision medicine. I have one disclosure to mention. And here's the basic concept that really we just heard about, the notion of cancer precision medicine, whereby a patient would have a biopsy tissue, could be a blood biopsy. It's subjected to some kind of omic analysis, and based on that genomic analysis, a clinician or the patient or some combination can then infer what are the right drugs to give, and the patient gets the drugs and enjoys a durable response, if not cure. Now, at the level of a PowerPoint slide, this works every time. The challenge is that the reality is that most cancer patients don't benefit from this kind of rubric. And I believe that this is not a fad. This is not just a theory or a concept. This has the potential to be the reality for all patients. And so we need to worry a lot about what's needed between where we are today and a future in which this is routine for all patients. Of course, getting the tumor genome is becoming increasingly routine. That's not the hard part. The hard part is figuring out what it means for therapeutic intervention. And so the two topics that I want to touch on here are here in red. I believe that we need a much stronger basic science foundation, preclinical foundation, from which we can interpret what the genome is telling us with respect to appropriate therapeutic interventions for patients. There aren't enough patients who are receiving enough treatments on the planet to be able to figure this out just from the patient experience, at least if we feel some urgency, as we should. We need to figure this out in large measure preclinically so that the appropriate clinical trials are prioritized rather than doing those trials randomly. And yet, there is a lot to be learned from the patient experience and treatments that patients are getting now. And most of that experience is lost into the ether somewhere, as we've been hearing about at this meeting, and we need better ways to capture that patient experience and learn from it. And so I'll talk about that as well. So let's start first focusing on preclinical models to predict vulnerabilities, or what I'm going to call cancer dependencies. The genes in the genome on which a tumor is dependent for survival, or the drugs that will kill a tumor selectively. And so at the Broad Institute, we've created a large-scale project that we are referring to as a cancer dependency map. That is a map of the breadth of the genetic heterogeneity of cancer, from which we can learn what are the dependencies or the vulnerabilities of tumors given their genome. This involves creating a large collection of cancer models, largely cell lines, that not just your favorite workhorse cancer cell line, but a diversity of models that reflects the molecular diversity of actual human cancer. Of course, those need to be systematically characterized at the genomic level, the details of which omic data are collected, I'll say a bit more about later. We then need to subject those to comprehensive drug sensitivity testing and genetic loss of function, which we can now do with genome editing techniques such as CRISPR-Cas9. And then from these data of genomically characterized cancer cell models, drug and genetic sensitivity testing, we can make a map of these cancer dependencies that are coupled to molecular predictors of response to those interventions. This should yield a blueprint for new targets that should be the subject of next generation drug development, and also the development of predictors, biomarkers of patient response to those particular therapeutic interventions. So where are we in trying to realize this idea? To do this, of course, if you want to represent the enormous molecular heterogeneity of human cancer, you need to do this at an appropriate scale. We think that in the end, the scale that's needed is probably going to be around 10,000 different models. And here's where we are at the moment, where we've got about 500 of those done so far. So 500 cancer cell lines that have been systematically subjected to multiple omic characterizations, as shown here. And then these have been subjected to 3 different types of manipulations. Knocking down essentially all genes in the genome by RNA interference. This is the first phase of the project. RNAi is well known to be plagued with off-target effects. On the other hand, it gives you partial loss of function of genes in a way that CRISPR-Cas9 knockout, while cleaner, gives you complete loss of function, which may not be the best way to model what a drug might do. And so we're actually finding that there's value in having both types of data. So RNAi partial knockdown, CRISPR-Cas9 knockout of all genes across these 500 cell lines, and then treatment of these roughly 500 cell lines with 5,000 drugs, FDA-approved drugs and drugs that are in clinical development, not necessarily for cancer, but for anything. Now, I think most people probably know that there have been methods developed for doing pooled RNAi or CRISPR knockout screens where you can take the whole genome as a pool and treat a cell line and figure out what the vulnerabilities are. But how do you approach this effectively for drugs where you can't pool all the drugs together? How do you do this in a rapid and cost-effective kind of way? And to do this, we've developed a method in the lab, and this is the work of Channing Yu, published about 2 years ago now, that we call PRISM, and I want to mention this briefly. The concept here is to take multiple different cancer cell lines, introduce a DNA barcode into each of them, which then allows you to pool the cell lines together, subject it to drug treatment, the pool to drug treatment, and then simply enumerate the abundance of the barcode before and after treatment, and thereby infer which of the cell lines in the pool were killed by the drug and which ones were not. We can do this either by a hybridization method to anti-barcode tags coupled to Luminex beads, or by next-generation sequencing. And for those that are interested in the details, we do this either as a pool of 25 cell lines per well of a 384 well plate, or around 600 cell lines in a single flask. Okay, all barcoded, all in a single pool. Here's just the proof of concept experiment that shows what we did in the first pool of 25 non-small cell lung cancer cell lines. Each, again, barcoded. Each cell line is a different column, and I'll just draw your attention here as an example. Here are the EGFR mutant cell lines, as shown here in orange. And here's a dose response to 3 different drugs, an EGFR inhibitor, which would be expected to kill only the EGFR mutant lung cancers. And you see, with killing shown in a blue color, in a dose response fashion, you see selective killing only of the EGFR mutant cell lines. So that's the concept, which we've now scaled to a scale of around 500 or 600 cell lines. Just a note about putting together a collection of interesting drugs to study. You would think that one could go to a definitive source and simply buy a collection of all FDA-approved drugs or drugs in clinical development. No such single source exists, and even the annotation for what these drugs are doesn't exist. And so we had to manually curate this and go to now over 75 different chemical vendors to obtain these different compounds, and importantly, validate that the compounds were what the vendors said they were. And here, a bit of a shock to those, at least the non-chemists in the room, certainly is a bit of a shock to me, was that 28% of the compounds failed quality quality control by mass spectrometry, either based on purity or identity of the compounds. Now, chemists would do this kind of checking routinely because they have learned that, yeah, that's kind of what you expect, and so why would you ever do a biology experiment without first doing that? That's not part of the normal routine of most biology labs, and it probably should be, because I'm not sure that this number is misrepresentative of most situations. Okay. But then we have a high-quality library in hand, and one can do interesting experiments like this. For example, treat 578 cancer cell lines in the PRISM format with the first 4,150 drugs. And you get a heat map that looks like this, again, where each cell line is a row and each drug is a column. And I won't go through this in detail except to say that, of course, you see that some compounds at a high concentration will kill everything. That's not shocking, and are probably not interesting, at least at that concentration. Many do absolutely nothing, and that's not shocking either because most of the drugs in the collection are not oncology drugs, and so one would not expect them to do anything. But you see some pockets of selective killing of certain compounds of a subset of cancer cell lines, and that's what's most interesting. One can look at this in a reduced dimensionality projection, the so-called TSNE projection, where one can see that different compounds cluster together in this TSNE analysis based on their mechanism of action. So, for example, all of the EGFR inhibitors cluster together in the space here, indicating that they have a similar mechanism of action. One can use this, of course, so it's not the subject of the talk today, to discover new compounds, unannotated compounds, that if you had a new compound that fell in this cluster, one could infer that it must be an EGFR inhibitor simply on the basis of its pattern of killing across 500 cancer cell lines. So what are the kinds of dependencies that we're looking for in this cancer dependency map? And so what is illustrated here is a distribution of killing, either by knocking out a gene or treating with a drug, looking at that distribution across 578, or around 500 cancer cell lines. And here are three patterns that one would think about. Here's the distribution around a score of 0, where one would conclude that this gene is not essential for survival of cancer cell lines really at all. It follows a normal distribution centered on 0. Here's the pattern of what we call common essentials, that is, those genes that kill basically all cell lines, or a drug that kills all cell lines. Those aren't interesting either, because that's just going to give you toxicity with no evidence of selective killing of a particular tumor population. The interesting guys are these, which we call preferential dependencies, which there's a left tail of this distribution, again, largely centered on 0, but a small number of cell lines on which there's a unique dependency, sensitivity to a drug, or a vulnerability dependency on a particular gene in the genome. And that's what we want to focus on. And then, with those preferential dependencies in hand, one can get to the business of, for example, correlating a particular dependency with a genomic feature. So we have, over 60,000 features, for example, all genes in the genome and their expression, mutation, copy number, and so on. And we can then develop predictive models that will use these features to develop predictive models that would predict, given the genome, the existence of a dependency. That's what we need to be able to do for patients. Read out the genome, apply a predictive model, and predict a vulnerability or drug sensitivity. That's the concept. How many of these strong preferential dependencies exist? We found about 700 of them so far that are at least 6 standard deviations away from the mean. So these are quite outlier dependencies. And using the genomic information that we have available, which is quite extensive for these cell lines, we actually do a decent job at predicting about half of them. Some of them, we observe the dependency but can't predict, have no molecular predictor of what is it that causes that dependency. So that's actually quite a higher number than we've ever had before, and it's there because the scale of the dependency map is getting to the point where there's enough data to be able to find these relationships and build predictive models. So now let's turn to the question of, well, we have all this omic data from the cell lines, and we do a decent job at building predictive models to predict drug sensitivity or gene dependency. Given all that information, what types of genomic information are carrying the most value or information in these molecular predictors? And that's shown here. And maybe let's start on the right here. Here's mutation data. So single nucleotide variants or small indels. This is just the number of predictive models. Does a decent job. Copy number does a decent job, a bit better. Here's gene expression. Substantially more predictive models use the gene expression data compared to DNA level information. And when you add all the data together, you do even better still, but this also illustrates how much of the overall predictability of drug sensitivity or gene dependency comes from gene expression. Why is this relevant? Because over the last decade, we've had a pretty dramatic emphasis on DNA level measurements in the clinic, either in diagnostic laboratories in industry or in academic labs. And I think what this dependency map is teaching us is that we need to resurrect gene expression as a form of a clinical biomarker, because otherwise we're going to be missing a lot of predictability for our patients. Okay, I want to turn now to the clinical side more briefly. That resonates, I think, with this idea we need to learn more from the patient experience, and we need to do a better job of having patient-centered efforts to collect information from patients as partners in this research effort. As I think people are aware here in oncology, those of us in research focus on major cancer centers typically and the patients that are seen there and are treated on clinical trials, and that's all wonderful. But the vast majority of patients in this country are not enrolled in any clinical trial, and they're not even treated in major cancer centers. They're treated in community practice. And most of these, 85% of the patients across the country don't have the opportunity. No one ever asks them, would you like to contribute to research, to understand how to predict better therapies and so on. But the world is changing now, as we've been hearing at this conference, through numerous advances in technology, social media, cultural changes which are increasing patients' openness to sharing their information, and patients just feeling more empowered to control their destiny more and be less passive about this. And this is all conspiring in a very good way, I think, to change the landscape significantly. And so we've launched an initiative that we're calling Count Me In, where the concept here is that patients should be able to raise their hand and say, I should have the right and the ability to share my information, whether it's genomic information or clinical information, with whoever I want to. That's my right. It's not my doctor's right. It's not the hospital's right. It's not a company's right. It's my right as a patient to do this. And so we launched a number of projects to try to do this. And I think it is similar to what we've been hearing from Ono and others at this conference. The first, led by Nick Wagley at the Broad and the Dana-Farber, focuses on metastatic breast cancer, where there's been relatively less emphasis on learning from these patients than for newly diagnosed patients with non-metastatic disease. And Nick has worked with patient advocacy groups and with many breast cancer patients to develop an online presence whereby patients can undergo informed consent independent of any institution, all online consent and providing patient information and the ability to collect samples, whether it's blood biopsy or permission to get their tissue blocks. Interestingly, 98% of patients who have entered the site have provided patient information, including their clinical information in terms of response to therapy and diagnostic information, which has been remarkably accurate compared to the gold standard of the medical record, which it tends to be part of the culture, well, you can't really trust the quality of data that comes from patients. It actually turns out not to be true, particularly when you're talking about patients that are deeply engaged in their own disease. This allows you to do all kinds of interesting things, like ask about patients that have had extraordinary responses to therapy of different types. For example, through this collection, hundreds of patients that have had unusually durable responses to therapy to which most patients don't benefit. And these types of numbers, just to take my word for it here, are difficult to get in any kind of institutional kind of way. It's only possible because you could reach out across the country direct to patients to get these kinds of numbers, and Nick is working those up. We spoke about rare diseases already this morning. Angiosarcoma is a rare cancer, only 300 new cases a year in the United States. Could you use this kind of count me in approach to attract patient participants for a rare disease in a way that any center will just see 0 or 1 of these every year? Answer is yes. In the first 6 weeks of launching the angiosarcoma count me in project, 300 patients were available, and so there will be an angiosarcoma genome project that hasn't happened before. And just a few weeks ago, we launched, through the leadership of Ellie Van Allen, a prostate cancer count me in project as well, and this is accruing patients as well. And so what we're seeing is when you look at patient engagement as partners in these projects, across these 3 projects, we have patients from all 50 states, the majority of whom are not being treated in major cancer centers. We don't yet have the ethnic diversity that we think this kind of project has the ability to attract. It's still primarily Caucasian individuals, so we want to focus on more diversity in that regard, but really excited about this idea of patients as partners, not as a replacement for academic medical center-driven research, but as a complementary approach to it. So I want to return, this is my last slide, to this notion. I think this is the right way to think about cancer, including immunotherapy. Immunotherapy isn't another idea. It's just another modality that is equally precision medicine and genomic medicine oriented. This is the right kind of model. We just need to do more work to make this routine for all patients. So let me end by thanking the leadership of these 2 projects. The cancer dependency map, in particular Bill Hahn, Jesse Bohm, Aviad Cherniak, and Paquita Vasquez, and the Count Me In project, in particular led by Nick Wagley. Thank you very much.